A mirror neuron is a neuron that fires both when an animal acts and when the animal observes the same action performed by another. Thus, the neuron mirrors the behavior of the other, as though the observer were itself acting. Such neurons have been directly observed in primate species. Birds have been shown to have imitative resonance behaviors, and neurological evidence suggests the presence of some form of mirroring system. In humans, brain activity consistent with that of mirror neurons has been found in the premotor cortex, the supplementary motor area, the primary somatosensory cortex and the inferior parietal cortex. The function of the mirror system is a subject of much speculation. Many researchers in cognitive neuroscience and cognitive psychology consider that this system provides the physiological mechanism for the perception-action coupling. They argue that mirror neurons may be important for understanding the actions of other people, and for learning new skills by imitation. Some researchers also speculate that mirror systems may simulate observed actions, and thus contribute to theory of mind skills, while others relate mirror neurons to language abilities. Neuroscientists such as Marco Iacoboni have argued that mirror neuron systems in the human brain help us understand the actions and intentions of other people. In a study published in March 2005, Iacoboni and his colleagues reported that mirror neurons could discern if another person who was picking up a cup of tea planned to drink from it or clear it from the table. In addition, Iacoboni has argued that mirror neurons are the neural basis of the human capacity for emotions such as empathy. It has also been proposed that problems with the mirror neuron system may underlie cognitive disorders, particularly autism. However, the connection between mirror neuron dysfunction and autism is tentative, and it remains to be seen how mirror neurons may be related to many of the important characteristics of autism. Despite the excitement generated by these findings, to date, no widely accepted neural or computational models have been put forward to describe how mirror neuron activity supports cognitive functions such as imitation. There are neuroscientists who caution that the claims being made for the role of mirror neurons are not supported by adequate research. Discovery. In the 1980s and 1990s, Giacomo Rizzolotti was working with Giuseppe Di Pellegrino, Luciano Fadiga, Leonardo Fgassi, and Vittorio Gallis at the University of Parma, Italy. These neurophysiologists had placed electrodes in the ventral premotor cortex of the macaque monkey to study neurons specialized for the control of hand and mouth actions. For example, taking hold of an object and manipulating it. During each experiment, the researchers allowed the monkey to reach for pieces of food and recorded from a single neuron in the monkey's brain, thus measuring the neuron's response to certain movements. They found that some of the neurons they recorded from would respond when the monkey saw a person pick up a piece of food as well as when the monkey picked up the food. The discovery was initially sent to Nature but was rejected for its lack of general interest. A few years later, the same group published another empirical paper, discussing the role of the mirror neuron system in action recognition, and proposing that the human BRCA Euro1 registered trademark S region was the homologue region of the monkey ventral premotor cortex. While these papers reported the presence of mirror neurons responding to hand actions, a subsequent study by Ferrari Pierfrancesco and colleagues described the presence of mirror neurons responding to mouth actions and facial gestures. Further experiments confirmed that about 10% of neurons in the monkey inferior frontal and inferior parietal cortex have mirror properties and give similar responses to performed hand actions and observed actions. In 2002, Christian Kieses and colleagues reported that, in both humans and monkeys, the mirror system also responds to the sound of actions. Reports on mirror neurons have been widely published and confirmed, with mirror neurons found in both inferior frontal and inferior parietal regions of the brain. Recently, evidence from functional neuroimaging strongly suggests that humans have similar mirror neuron systems. Researchers have identified brain regions which respond during both action and observation of action. Not surprisingly, these brain regions include those found in the macaque monkey. However, functional magnetic resonance imaging can examine the entire brain at once and suggests that a much wider network of brain areas shows mirror properties in humans than previously thought. These additional areas include the somatosensory cortex and are thought to make the observer feel what it feels like to move in the observed way. Origin. The most common theory behind the origin of mirror neuron is the genetic account, which suggests that the mirrorness of mirror neurons is due primarily to heritable genetic factors and that the genetic predisposition to develop mirror neuron evolved because they facilitate action understanding. The other theories as to the origin of mirror neurons include associative learning, canalization and exorptation. In monkeys. The first animal in which researchers have studied mirror neurons individually is the macaque monkey. In these monkeys, mirror neurons are found in the inferior frontal gyrus and the inferior parietal lobule. Mirror neurons are believed to mediate the understanding of other animals' behavior. For example, a mirror neuron which fires when the monkey rips a piece of paper would also fire when the monkey sees a person rip paper, or hears paper ripping. These properties have led researchers to believe that mirror neurons encode abstract concepts of actions like ripping paper, whether the action is performed by the monkey or another animal. The function of mirror neurons in macaques remains unknown. Adult macaques do not seem to learn by imitation. Recent experiments by Ferrari and colleagues suggest that infant macaques can imitate a human's face movements, though only as neonates and during a limited temporal window. Even if it has not yet been empirically demonstrated, it has been proposed that mirror neurons underlie this behavior and other imitative phenomena. Indeed, there is limited understanding of the degree to which monkeys show imitative behavior. In adult monkeys, mirror neurons may enable the monkey to understand what another monkey is doing, or to recognize the other monkey's action. In humans, it is not normally possible to study single neurons in the human brain, so most evidence for mirror neurons in humans is indirect. Brain imaging experiments using functional magnetic resonance imaging have shown that the human inferior frontal cortex and superior parietal lobe are active when the person performs an action and also when the person sees another individual performing an action. It has been suggested that these brain regions contain mirror neurons, and they have been defined as the human mirror neuron system. More recent experiments have shown that even at the level of single participants, scanned using fMRI, large areas containing multiple fMRI voxels increase their activity both during the observation and execution of actions. Neuropsychological studies looking at lesion areas that cause action knowledge, pantomime interpretation, and biological motion perception deficits have pointed to a causal link between the integrity of the inferior frontal gyrus and these behaviors. Transcranial magnetic stimulation studies have confirmed this as well. These results indicate the activation in mirror neuron-related areas are unlikely to be just epiphenomenal. A study published in April 2010 reports recordings from single neurons with mirror properties in the human brain. Macaques et al. Recorded from the brains of 21 patients who were being treated at Ronald Reagan UCLA Medical Center for intractable epilepsy. The patients had been implanted with intracranial depth electrodes to identify seizure foci for potential surgical treatment. Electrode location was based solely on clinical criteria. The researchers, with the patient's consent, used the same electrodes to piggyback their research. The researchers found a small number of neurons that fired or showed their greatest activity both when the individual performed a task and when they observed a task. Other neurons had anti-mirror properties, that is, they responded when the participant performed an action but were inhibited when the participant saw that action. The mirror neurons found were located in the supplementary motor area and medial temporal cortex. For purely practical reasons, these regions are not the same as those in which mirror neurons had been recorded from in the monkey. Researchers in Parma were studying the ventral premotor cortex and the associated inferior parietal lobe, two regions in which epilepsy rarely occurs, and hence, single-cell recordings in these regions are not usually done in humans. On the other hand, no one has to date looked for mirror neurons in the supplementary motor area or the medial temporal lobe in the monkey. Together, this therefore does not suggest that humans and monkeys have mirror neurons in different locations, but rather that they may have mirror neurons both in the ventral premotor cortex and inferior parietal lobe, where they have been recorded in the monkey, and in the supplementary motor areas and medial temporal lobe, where they have been recorded from in human especially because detailed human fMRI analyses suggest activity compatible with the presence of mirror neurons in all these regions. Another study has suggested that human beings don't necessarily have more mirror neurons than monkeys, but instead that there is a core set of mirror neurons used in action observation and execution. However, for other proposed functions of mirror neurons, the mirror system may have the ability to recruit other areas of the brain when doing its auditory, somatosensory, and affective components. Doubts concerning mirror neurons. Although many in the scientific community have expressed excitement about the discovery of mirror neurons, there are scientists who have expressed doubts about both the existence and role of mirror neurons in humans. According to scientists such as Hickok, Pascalot, and Dinstein, it is not clear whether mirror neurons really form a distinct class of cells, and whether mirror activity is a distinct type of response or simply an artifact of an overall facilitation of the motor system. In 2008, Elan Dinstein al. argued that the original analyses were unconvincing because they were based on qualitative descriptions of individual cell properties, and did not take into account the small number of strongly mirror-selective neurons in motor areas. Other scientists have argued that the measurements of neuron fire delay seem not to be compatible with standard reaction times, and pointed out that nobody has reported that an interruption of the motor areas in F5 would produce a decrease in action recognition. In 2009, Ling al. carried out an experiment in which they compared motor acts that were first observed and then executed to motor acts that were first executed and then observed. They concluded that there was a significant asymmetry between the two processes that indicated that mirror neurons do not exist in humans. They stated crucially, "We found no signs of adaptation for motor acts that were first executed and then observed." Failure to find cross-modal adaptation for executed and observed motor acts is not compatible with the core assumption of mirror neuron theory, which holds that action recognition and understanding are based on motor simulation. However, in the same year, Kilner al. showed that if goal-directed actions are used as stimuli, both IPL and premotor regions show the repetition suppression between observation and execution that is predicted by mirror neurons. In 2009, Greg Hickok published an extensive argument against the claim that mirror neurons are involved in action understanding, 8 problems for the mirror neuron theory of action understanding in monkeys and humans. He concluded that the early hypothesis that these cells underlie action understanding is likewise an interesting and prima facie reasonable idea. However, despite its widespread acceptance, the proposal has never been adequately tested in monkeys, and in humans there is strong empirical evidence, in the form of physiological and neuropsychological dissociations, against the claim. Vladimir Kosnagov sees another contradiction. The proponents of mirror neuron theory of action understanding postulate that the mirror neurons code the goals of others' actions because they are activated if the observed action is goal-directed or is a pantomime of a goal-directed action. However, the mirror neurons are activated only when the observed action is goal-directed or is a pantomime of a goal-directed action. How do they know that the definite action is goal-directed or is a pantomime of the goal-directed action? At what stage of their activation do they detect a goal of the movement or its absence? In his opinion, the mirror neuron system can be activated only after the goal of the observed action is attributed by some other brain structures. Neurophilosophers such as Patricia Churchland have expressed both scientific and philosophical objections to the theory that mirror neurons are responsible for understanding the intentions of others. In Chapter 5 of her 2011 book, Brain Trust, Churchland points out that the claim that mirror neurons are involved in understanding intentions is based on assumptions that are clouded by unresolved philosophical issues. She makes the argument that intentions are understood at a more complex level of neural activity than that of individual neurons. Churchland states that a neuron, though computationally complex, is just a neuron. It is not an intelligent homunculus. If a neural network represents something complex, such as an intention, to insult, it must have the right input and be in the right place in the neural circuitry to do that. Recently, Cecilia Hayes has advanced the theory that mirror neurons are the byproduct of associative learning as opposed to evolutionary adaptation. She argues that mirror neurons in humans are the product of social interaction and not an evolutionary adaptation for action understanding. In particular, Hayes rejects the theory advanced by versus Ramachandran that mirror neurons have been the driving force behind the great leap forward in human evolution. Development. Human infant data using eye tracking measures suggest that the mirror neuron system develops before 12 months of age, and that this system may help human infants understand other people's actions. A critical question concerns how mirror neurons acquire mirror properties. Two closely related models postulate that mirror neurons are trained through Hebbian or associative learning. However, if premotor neurons need to be trained by action in order to acquire mirror properties, it is unclear how newborn babies are able to mimic the facial gestures of another person, as suggested by the work of Meltzoff and Moore. One possibility is that the site of tongue protrusion recruits an innate releasing mechanism in neonates. Careful analysis suggests that imitation of this single gesture may account for almost all reports of facial mimicry by newborn infants. Possible functions: understanding intentions. Many studies link mirror neurons to understanding goals and intentions. Gassi al. recorded the activity of 41 mirror neurons in the inferior parietal lobe of two rhesus macaques. The IPL has long been recognized as an association cortex that integrates sensory information. The monkeys watched an experimenter either grasp an apple and bring it to his mouth or grasp an object and place it in a cup. In total, 15 mirror neurons fired vigorously when the monkey observed the grasp-to-eat motion, but registered no activity while exposed to the grasp-to-place condition. For 4 other mirror neurons, the reverse held true. They activated in response to the experimenter eventually placing the apple in the cup but not to eating it. Only the type of action, and not the kinematic force with which models manipulated objects, determined neuron activity. It was also significant that neurons fired before the monkey observed the human model starting the second motor act. Therefore, IPL neurons code the same act in a different way according to the final goal of the action in which the act is embedded. They may furnish a neural basis for predicting another individual unregistered trademark as subsequent actions and inferring intention. Learning facilitation. Another possible function of mirror neurons would be facilitation of learning. The mirror neurons code the concrete representation of the action, that is, the representation that would be activated if the observer acted. This would allow us to simulate the observed action implicitly to collect our own motor programs of observed actions and to get ready to reproduce the actions later. It is implicit training. Due to this, the observer will produce the action explicitly with agility and finesse. This happens due to associative learning processes. The more frequently a synaptic connection is activated, the stronger it becomes. Empathy. Stephanie Preston and Franz de Waal, Jean Dessuty, and Vittorio Gallis and Christian Kieses have independently argued that the mirror neuron system is involved in empathy. A large number of experiments using fMRI, electroencephalography and magnetoencephalography have shown that certain brain regions are active when people experience an emotion and when they see another person experiencing an emotion. However, these brain regions are not quite the same as the ones which mirror hand actions, and mirror neurons for emotional states or empathy have not yet been described in monkeys. More recently, Christian Kieses at the Social Brain Lab and colleagues have shown that people who are more empathic according to self-report questionnaires have stronger activations both in the mirror system for hand actions and the mirror system for emotions, providing more direct support for the idea that the mirror system is linked to empathy. Some researchers observed that the human mirror system does not passively respond to the observation of actions but is influenced by the mindset of the observer. Researchers observed the link of the mirror neurons during empathetic engagement in patient care. Human self-awareness versus Ramachandran has speculated that mirror neurons may provide the neurological basis of human self-awareness. In an essay written for the Edge Foundation in 2009, Ramachandran gave the following explanation of his theory. I also speculated that these neurons can not only help simulate other people's behavior but can be turned inward as at array to create second-order representations or meta-representations of your own earlier brain processes. This could be the neural basis of introspection, and of the reciprocity of self-awareness and other awareness. There is obviously a chicken or egg question here as to which evolved first, but the main point is that the two co-evolved, mutually enriching each other to create the mature representation of self that characterizes modern humans. Language. In humans, functional MRI studies have reported finding areas homologous to the monkey mirror neuron system in the inferior frontal cortex, close to Broca's area, one of the hypothesized language regions of the brain. This has led to suggestions that human language evolved from a gesture performance understanding system implemented in mirror neurons. Mirror neurons have been said to have the potential to provide a mechanism for action understanding, imitation learning, and the simulation of other people's behavior. This hypothesis is supported by some Sitka tectonic homologies between monkey premotor area F5 and human Broca's area. Rates of vocabulary expansion linked to the ability of children to vocally mirror non-words and so to acquire the new word pronunciations. Such speech repetition occurs automatically, fast and separately in the brain to speech perception. Moreover, such vocal imitation can occur without comprehension, such as in speech shadowing and echolalia. Further evidence for this link comes from a recent study in which the brain activity of two participants was measured using fMRI while they were gesturing words to each other using hand gestures with a game of charades, a modality that some have suggested might represent the evolutionary precursor of human language. Analysis of the data using Granger causality revealed that the mirror neuron system of the observer indeed reflects the pattern of activity in the motor system of the sender, supporting the idea that the motor concept associated with the words is indeed transmitted from one brain to another using the mirror system. The mirror neuron system seems to be inherently inadequate to play any role in syntax, given that this definitory property of human languages which is implemented in hierarchical recursive structure is flattened into linear sequences of phonemes, making the recursive structure not accessible to sensory detection, automatic imitation. The term is commonly used to refer to cases in which an individual, having observed a body movement, unintentionally performs a similar body movement or alters the way that a body movement is performed. Automatic imitation rarely involves overt execution of matching responses. Instead, the effects typically consist of reaction time, rather than accuracy, differences between compatible and incompatible trials. Research reveals that the existence of automatic imitation, which is a cover form of imitation, is distinct from spatial compatibility. It also indicates that, although automatic imitation is subject to input modulation by attentional processes, and output modulation by inhibitory processes, it is mediated by learned, long-term sensorimotor associations that cannot be altered directly by intentional processes. Many researchers believe that automatic imitation is mediated by the mirror neuron system. Additionally, there are data that demonstrate that our postural control is impaired when people listen to sentences about other actions. For example, if the task is to maintain posture, people do it worse when they listen to sentences like this: I get up, put on my slippers, go to the bathroom. This phenomenon may be due to the fact that during action perception there is similar motor cortex activation as if a human being performed the same action. Motor mimicry. In contrast with automatic imitation, motor mimicry is observed in naturalistic social situations and via measures of action frequency within a session rather than measures of speed and/or accuracy within trials. The integration of research on motor mimicry and automatic imitation could reveal plausible indications that these phenomena depend on the same psychological and neural processes. Preliminary evidence, however, comes from studies showing that social priming has similar effects on motor mimicry. Nevertheless, the similarities between automatic imitation, mirror effects, and motor mimicry have led some researchers to propose that automatic imitation is mediated by the mirror neuron system and that it is a tightly controlled laboratory equivalent of the motor mimicry observed in naturalistic social contexts. If true, then automatic imitation can be used as a tool to investigate how the mirror neuron system contributes to cognitive functioning and how motor mimicry promotes prosocial attitudes and behavior. Meta-analysis of imitation studies in humans suggests that there is enough evidence of mirror system activation during imitation that mirror neuron involvement is likely, even though there are no published studies have recorded the activities of singular neurons. However, it is likely insufficient for motor imitation. Studies show that regions of the frontal and parietal lobes that extend beyond the classical mirror system are equally activated during imitation. This suggests that other areas, along with the mirror system, are crucial to imitation behaviors. Autism. Some researchers claim there is a link between mirror neuron deficiency and autism. EEG recordings from motor areas are suppressed when someone watches another person move, a signal that may relate to mirror neuron system. This suppression was less in children with autism. Although these findings have been replicated by several groups, other studies have not found evidence of a dysfunctional mirror neuron system in autism. In 2008, Iacoboni et al. Published a research paper that presented conflicting EEG evidence. Iacoboni and Ramachandran found typical mu suppression for familiar stimuli, but not for unfamiliar stimuli, leading them to conclude that the mirror neuron system of children with ASD was functional, but less sensitive than that of typical children. Based on the conflicting evidence presented by mu-wave suppression experiments, Patricia Churchland has cautioned that mu-wave suppression results cannot be used as a valid index for measuring the performance of mirror neuron systems. Finally, anatomical differences have been found in the mirror neuron-related brain areas in adults with autism spectrum disorders, compared to non-autistic adults. All these cortical areas were thinner and the degree of thinning was correlated with autism symptom severity, a correlation nearly restricted to these brain regions. Based on these results, some researchers claim that autism is caused by impairments in the mirror neuron system, leading to disabilities in social skills, imitation, empathy and theory of mind. Many researchers have pointed out that the broken mirrors theory of autism is overly simplistic, and mirror neurons alone cannot explain the deficits found in individuals with autism. First of all, as noted above, none of these studies were direct measures of mirror neuron activity, in other words fMRI activity or EEG rhythm suppression do not unequivocally index mirror neurons. Dinstein and colleagues found normal mirror neuron activity in people with autism using fMRI. In individuals with autism, deficits in intention understanding, action understanding and biological motion perception are not always found, or are task-dependent. Today, very few people believe an all-or-nothing problem with the mirror system can underlie autism. Instead, additional research needs to be done, and more caution should be used when reaching out to the media. Theory of mind. In philosophy of mind, mirror neurons have become the primary rallying call of simulation theorists concerning our theory of mind. Theory of mind refers to our ability to infer another person's mental state from experiences or their behavior. For example, if you see a girl reaching into a jar labeled cookies, you might assume that she wants a cookie and believes that there are cookies in the jar. There are several competing models which attempt to account for our theory of mind. The most notable in relation to mirror neurons is simulation theory. According to simulation theory, theory of mind is available because we subconsciously empathize with the person we're observing and, accounting for relevant differences, imagine what we would desire and believe in that scenario. Mirror neurons have been interpreted as the mechanism by which we simulate others in order to better understand them, and therefore their discovery has been taken by some as a validation of simulation theory. More recently, theory of mind and simulation have been seen as complementary systems, with different developmental time courses. Gender differences. A series of recent studies conducted by Yawei Cheng, using a variety of neurophysiological measures, including MEG, spinal reflex excitability, electroencephalography, have documented the presence of a gender difference in the human mirror neuron system, with female participants exhibiting stronger motor resonance than male participants. In another study, gender differences among mirror neuron mechanisms was reinforced and that the data showed enhanced empathetic ability in females when compared to males. During an emotional social interaction, females show a greater ability in emotional perspective taking than do males when interacting with another person face-to-face. This ability may be due to the fact that females express their own emotion more thoroughly than males do. However, in the study, data showed that when it came to recognizing the emotions of others, both males and females' ability was very similar and there was no key difference between genders. See also, associative sequence learning, common coding theory, emotional contagion, empathy, motor cognition, motor theory of speech perception, on intelligence, positron emission tomography, simulation theory of empathy, speech repetition, spindle neuron, references. Further reading, Keysers, see The Empathic Brain or, Iacoboni M., Mazziota J.C. Mirror Neuron System, Basic Findings and Clinical Applications. And Neural 62, 213 8 doi, 10.1002/ANA 21198. PMIDA 17721988 Keysers, C., and Gazzola, V., Towards a Unifying Neural Theory of Social Cognition, Progress in Brain Research. 1, Mazur, E., Bug, J.A., and Gollwitzer, P.M. Oxford Handbook of Human Action. New York, Oxford University Press. Preston, S.D., and DeWall, F.B.M. Empathy, Its Ultimate and Proximate Bases. Behavioral and Brain Sciences, 25, 1-72. Rizzolotti G., Fabridistro M., Cattaneo L. Mirror Neurons and Their Clinical Relevance. Nat Clin Pract Neural 5, 24 €34 doi, 10.1038/UNCP Neuro 0990. PMIDA 19129788 Rizzolotti, G., Sinigaglia, C. Mirrors in the Brain. How We Share Our Actions and Emotions. Oxford University Press. External links, Neuroscience Now, Mirror Neurons, You Remind Me of Me in the New York Times. 7-minute video on TED.com. A neuroscientist Vilayana Ramachandran outlines the functions of mirror neurons. Mirror Neuron Forum, Perspectives on Psychological Science, September, 2011, Talking Brains, Greg Hickok and David Poepel, News and Views on the Neural Organization of Language, Thomas, Ben, Euro Unregistered Trademark So Special About Mirror Neurons? Scientific American Guest Blog, 2012, 2.